Muhammad Umar Farooq biologically video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. Answer question to each other. So here we will discuss about the gene mutation. So overall this diagram we will understand the gene mutation throughout the video. So the different type of gene mutation we will discuss. So here just we will focus on the concept of the gene. So here is the cell, is the somatic cell. For example, in our body, which that is able to divide. So that's contain nucleus, and this nucleus contain are different type of chromosomes. So the 46 chromosome in the somatic cell in our body in the form of pair. So that chromosome is made up of DNA. So the DNA is the basic structural and functional unit of the gene, which that is expressed the genotype into the phenotype. So these genotype, basically this DNA is wind with the histone protein. So the 8 subunit of the histone protein H2A, H2B, H2B, and H4 as well as H3 and H1, which that is the monomer, and that is whole diagram is known as the nucleosome. And this nucleosome is contain one bead of the histone protein is 10 nanometer, while the total size of this is the 30 nanometer, while here contain basically 100 and 106 base pair of DNA which contain a nucleosome. So this is 8 histone protein monomer. So that's contain DNA. DNA is the double stranded nucleotides. That is the basically macromolecule which that are present and contain a gene. For example, expression of the gene 1 is a separate, while G2, G3, G4 and G5 which that express separately. So anyhow, here we will discuss the mutation. What is the mutation? Mutation, the change in the genetic makeup of a cell, are called mutation. So the mutation can be harmful or helpful. For example, in the plant which that is the helpful. So that is the alter the structure of chromosome, and that is the point mutation in gametes or in a cell transmit to offspring. So that sperm and egg contain a mutation if so that is transferred into the offspring, which that is the genetically differentiation. So for example, the example is the sickle cell anemia is the point mutation. Anyhow, the Turner syndrome, Down syndrome, chromosomal mutation is also occur, which that is the Down syndrome and Klinefelter syndrome. We will discuss in later in the next video. So later on we will discuss also the mutagenesis. So first of all we should focus on the, here is the DNA which contain a nitrogenous bases. So the thymine, cytosine, thymine, adenine, guanine. So the thymine already must be attached with the adenine with the hydrogen bonding. So the double-stranded RNA double-stranded structure is looking like this. So that is the point mutation is due to the one change of the codon. So the codon is the responsible for the production of one amino acid from one codon. So that's the sickle cell anemia is the point mutation, which that is showing in the figure. But that is the nucleotide. For example, cytosine, guanine, and thymine and adenine. There's a 4 nitrogenous bases throughout the DNA. So the DNA is the long thread-like structure are present in the cell, which that's condensed in the form of chromosome during the cell division, so that we can easily understand the chromosome. So the type of mutation here we will discuss a base pair substitution. Substitution mean the changing. So the missense mutation, which that's contain a harmless missense mutation and lethal missense mutation. Lethal mean it is harmful, while the missense mutation harmless is also, and nonsense mutation is also the part of the base pair substitution. While here the base pair in insertion and deletion or frame shift mutation is also occur. So this type of insertion, addition, deletion loses nucleotides. One or more nucleotides is that can be happen. So let's talk about the base pair substitution. It means the base pair will be convert or change. So how it's possible? So the original DNA for code for an amino acid sequence will be changed. It means the one base pair will be changed to change the one codon which that is specific for the production of amino acid. So let's talk about here is the codon CAT, cytosine, adenine and thymine, which that is opposite to guanine, thymine and adenine. So that means the original DNA for code for an amino acid sequence is here, which produce the CAT, which that is produce the codon with the binding of the anticodon during the messenger RNA production to produce the anticodon, which are GTA, and that's produce the histamine, that is the histamine protein amino acid. This histamine is the polypeptide formation, and this polypeptide is known as the protein. So this protein formation, whenever that is changing of the one base pair to alter the gene expression. So the replacement of single nucleotides we will discuss in this diagram. So let's talk about the changing or replacement of a single nucleotide to alter the mechanism of the translation during the transcription after the translation, which that produce the protein. So the translation occur in the cytoplasm while the transcription occur in the nucleus. So here is the one base pair will be changed to alter the one codon also. So that is replacement of single nucleotide to change the codon. So that means the guanine attached with the cytosine is that one base pair which that will be altered to produce proline. So this proline will be inserted in the it is missense mutation. Missense mutation is the incorrect amino acid. So for example, it will be harmless or lethal missense mutation. So that's a malfunctioning protein will be occur, and that is not good correct functionally. While here the missense mutation, which that is the first we will discuss about the harmless mutation. It means the no effect to the generally physiological effect will be not occur. So that change of the one amino acids, no effect and normal function of the protein in the cellular either or in the structural you can understand. So lethal missense mutation can be impair cellular function. For example, the changing of the one nucleotide in the sickle cell anemia is due to this one. So that is the lethal missense because the hemoglobin trapping into the red blood cell is impaired. So that means the impaired cellular function also. Right? I hope you understand about the base pair substitution and replacement of single nucleotide while missense mutation are harmless. So now we will discuss about the nonsense mutation. So the nonsense mean the change of one total codon to change. For example, here is the tissue which contain DNA in the nucleus during the transcription that produce the messenger RNA. After the splicing, intron will be excreted while the exon will left behind, and that produce the 5' cap and poly A tail. So that means the adenine will be in the form of tail. So here is the different codon which that produce the normally that's protein, methionine, lysine, phenyl, alanine, glycine, alanine, leucine and glycine. That is the sequence of the protein production during the translation from the transfer RNA attachment with the ribosome. So, for example, AAG will be converted into the UAG. So, this UAG is the stop codon. So, this UAG will be stopped. It means after the production of methionine and lysine, that will be during the lysine, that will be codon stop. So, this stop codon produce the methionine just and stop. So, that means it is nonsense mutation. Nonsense mutation mean the stop of the codon the stop codon will be occur by the alteration of the gene nucleotide. So that means that malfunction or basically impair of the cellular function or something. So the base pair insertion and deletion or frame shift mutation is the second one, which that means the base pair is completely shift or inverse we will discuss. So the insertion and deletion or addition or losses of one or more nucleotides pair in a gene. This mutation a more dangerous effect. For example, here we will discuss that is the messenger RNA during the transcription that's produced and translation during the one uracil will be excrete or delete to produce this type of protein. So this type of protein mean that is the basically, for example, here is the normally that methionine, lysine, phenyl, alanine and glycine and stop. But here is the basically methionine, lysine and leucine and alanine will be formed. That is the totally change. And that means the function of the protein during the, for example, muscle synthesis or something is impaired. And that means the loss of the normal activity. So that means here the methionine, lysine, phenyl, alanine, glycine, that is after the stop. But here is a different, and that is stop is also occur. So that means the deletion effect are largely in the messenger RNA, while in the DNA basically, while in the addition if we will talk about, so the addition can be delete or something, addition can be stop the codon by the UAG. So that means methionine after translation to stop, so this stop codon, while here is the inversion. So the inverse of the nitrogenous base nucleotide basically, so the inversion to stop codon also. So that means the base pair insertion and deletion or frame shift mutation will be occur. So next here we will discuss about the mutagenesis. So the mutagenesis means the creation of mutation is called mutagenesis. So that's mutagenesis is the process which that create the mutation. So the mutation here we will discuss about the first is the major is the waves. So this wave direction is here and that's amplitude, and here is the lambda which that's crust and trough are present. So that is the physics topic, but we will just understand. So this is the mutagens, for example radiation, chemical or something other or toxins. So mutagen is a physical or chemical agents which affect to the mutation. So the effect to the mutation due to the mutagens. So the mutagen can be chemicals, toxin, acids, metal ions or something. So that effect in our body to change the DNA, and that will be the DNA damage. It means the alteration of the gene to produce a new type of protein which that is the harmful for us. So that means, for example, the UV light which that affect in our skin to change the DNA. So that means it change the DNA or activate the DNA to produce more melanin pigment. So that means it change the DNA sequence, while it is also damage the DNA to become a cancerous cell due to the chromosomal damage. So that means the DNA will be damaged to after the cell division to incorrect cell division will be occur. So that is why. Or on the other X-ray or microwave is also affecting our body to change the DNA. So that is the mutagens, and that's mutagen create the mutagenesis. While here the X-ray, UV light, other toxin is basically occur. So here is the plant which that is the harmless mutation occur in plants, and the mutation by the insertion of gene by biotechnology with our own selection to change the food product. So that is plant is important for the harmless mutation will be occur to change the growth of the plant. While on the other hand, epigenetics on the basis of DNA modification is also occur, which that is the change of the nucleotide to use as adaptation modification, resistance, age-wise activation, and expression of gene on the basis of requirement. For example, on the basis of environmental changes which that affect and activate the gene is known as the epigenetics. On the basis of basically epigenetic is a throughout our body is used as epigenetic for the adaptation, while the fertilization is the genetics.